so I'm going to be primarily talking about Flywheel and some of the efforts that we currently have in structuring imaging data, preparing it for analysis like semi or automated segmentation for CBTN data in particular, as well as how we're addressing some of the barriers in technology and computation. So processing radiology data originally looked like this. It required programming language knowledge. Luckily, that didn't last too long, and about like 30 years ago, people and groups started developing standalone software tools that were open source, free to download off the internet and use locally. And there are a lot of features in these types of softwares that are common, that are required for performing imaging analysis. As you can see, they all have viewers. They all have built-in ways to essentially manipulate the images. They have a set of standard analyses and processes that can be run on those images, but also allow users to custom define and manually tune some of those tools. So this is totally feasible to do for small data sets and is still, in a lot of cases, the kind of standard of data storage and processing in research labs and research teams to date. So usually people will store their data either on their local computer or on a like lab-owned or department-owned computing cluster, and then they'll perform their analysis. They'll publish their findings in scientific journals, granted that they have findings to publish, and sometimes they'll share their data and their code and their model files that they use to that they published. So this has also, happened and advanced with open science practices over the last like 10 or 20 years. But even if people share their data and their code, often they just throw it in a, separate siloed place, and that data and code becomes stale pretty fast. So it's not reproducible. It's not generalizable. You typically can't just pick up files and plug it in on your own system. And so with the progress of building out large-scale imaging data sets, there was a realized need, and a lot of groups quickly moved towards implementing cloud-based environments and using cloud services for their strong efficiency, their computational resources, as well as the development of platforms, software platforms that are specialized to analyze these specific data types. So whether it be Flywheel for imaging data or Cavatica for molecular and other data types, a lot of these platforms have user interfaces. They have built-in software tools and workflows. They allow user ease for data management, and they also allow reproducible coding and data standards that can be shared between groups. So just to underscore why this is extremely important, and when facing clinically collected imaging data, and apologies, the text here is a little small, but what I'm showing you here is essentially each column is a type of imaging sequence. So you can think of it as just a type of image. Each of these rows corresponds to what could come off the scanner, and this is based on real data from CBTN and PNOC. So each of these rows all fall under this category. But typically what's done is a researcher will collect all this data and be faced with figuring out for themselves which files based on these file names correspond to which of these types of images. So really, to empower researchers when you start dealing with 200,000 images is the need to be able to start grouping and classifying images and making it much more queryable and usable. So Flywheel has a lot of built-in ways to be able to handle cases like that. It allows secure access, auditing features. It has a built-in viewer, which I'll show you. There are query tools, automated classification of images, and also it allows built-in software that can either be shared between groups and community sourced, but it also allows people with programming knowledge to use their open Python toolkits and open REST APIs. All right, let's hope this works. All right, so here what I'm showing you is I've just logged into a sample user, and to log in essentially just requires a Gmail address. Once people have been granted access to a given data set, they would see that on their landing page here. So you can see each of the different data sets I've been granted access to and high level project or data set information. Then going into a given data set. All right, awesome. You can start to see for each subject the different exams collected and made available for those subjects. They are labeled by the agent days at the time of imaging, as well as the primary body part examined. And at the subject, session, and file level, there's a lot of flexible filtering that you can do. So for example, I can look for a specific subject. I can select a time point of interest. And on the right here, you can see the images acquired in that session. Go back to the top. So as you can see, the images have their description as it came off the scanner. So I can filter based on the file name, but I can also, using the automated labeling that's been done, look for a specific type of image. And this type of filtering can be done both at the level of a given subject that I'm looking at, but also there's search tools that can be used to look and query data across an entire data set or between data sets. All right, so let's see. So here I've just narrowed down to T1MP RAGE sequence. And if I look, for example, I've found one that I want to open. I can use just this built-in viewer. So as you can see, the toolbar at the top, there's a bunch of different features you can use. There's annotation tools and segmentation tools. I can also navigate between different time points for a given subject using this navigation panel. I can pull up images side by side across different exams or time points. I can scroll through the volume, and I can also make measurements and save them so that they can then again be aggregated across subjects and used at a higher level. There's ability for downloading images. You can download selected time points of interest through a drop-down menu. You can also download an entire project. And importantly, Flywheel captures at each level different metadata about the images collected and the subjects. So I have all of the metadata from the DICOMs that came off the scanner. I have the higher level classifications that have been automatically generated, and also users are empowered to create their own custom classifications and custom tags that again enables use at scale. All right, so that's a general gist. You can Flywheel has some upload capacities. It also has built-in software tools that can be used to operate on the images, where users can click through the AI, through the UI, and essentially just run through the user interface itself without any programming knowledge, standalone software tools. All right, so for the sake of time, I will jump back to the presentation. Let's see. Awesome. All right, so tools, again, like I mentioned earlier, can be input into a community sourced gear library, which is super important for being able to reproduce the analyses that are run on different data sets and across teams. And our team in particular is building out, as Miriam alluded to, pediatric specific processing pipelines that will essentially be applicable to the CBTN data set to reduce this extremely manual and time consuming steps of preparing the data to be input into model building. There's some work currently being done to assess and address methods of interoperability between platforms to allow users to link data across different data types between Flywheel and Cavatica in particular, and to be able to perform multimodal research. And again, all of this is deployed in the cloud. There's a lot of work for automation, which is super important when you're dealing with such a large data set.